Okay, so thank you for this kind introduction. I'm Charbel ELIAS from the Aix-Marseille University. I'm going to be attacking my subject directly. Don't want to hold you much longer, you guys. So, as you may know, fungi are key ecological actors in soil environments and have emerged during the past few decades as potential bioremediation and bioindicator agents. Yet our knowledge regarding fungal community structure remains relatively scarce when compared to bacteria. This gap in knowledge can be traced in part to the extended diversity of fungi, but also to the limitations associated with traditional and current methods used for this objective. So in this context, we'll be aiming at characterizing the soil microbiome or the soil community, fungal community structure, this time using a novel approach in addition to the ELIAS metabarcoding, which is currently used as a current standard for environmental sciences. We'll be using the Culturomics approach, which was initially and for the past decade used for the characterization of the human gut microbiota and led to the discovery of hundreds of novel bacterial species. So this time we'll be aiming at applying this approach for the soil communities, for the soil fungal community. So how do we proceed in this approach? Well, first of all, the soil samples are suspended in a DPVL solution, then serially diluted. Every dilution, along with the mother solution, are then plated onto 5 different culture media, among them some in-house culture media that we used for the first time in this approach. Then, after incubation, grown colonies are identified in the first place using the MALDI TOF mass spectrometry that we can see here. In fact, this technology generates a species-specific mass spectra that is compared to a reference database of mass spectra and then generates a score with the identity. Nonetheless, sometimes we aren't able to identify them with high scores because this technology is currently under, development for fungi. For bacteria, it works perfectly, but for fungi, it's not always the case, especially for environmental fungus. So in this case, we applied the Sanger sequencing, where we target, amplify, and sequence barcode regions, which in our case are the ITS sequences, the beta-tubulin gene, and the TOF genes. So using this approach, we were able to isolate, characterize, and identify 77 different fungal species belonging to 30 genera and 24 families. Analyzed soils were dominated at the family level by Aspergillaceae, followed by Nectariaceae, which translates at the genus level by an abundance in Aspergillus, followed by Penicillium and Fusariums. So we constructed a phylogenetic tree with the identified species, and when we dug a bit deeper, we saw that 3 among the species that we've isolated, Aspergillus iragawe, Aspergillus niger, and Curvularia inequalis, were more likely to be associated with a PAH polluted soil profile since they were present in all except one polluted profile, and at the same time, none present in all non-polluted soils. So, which make them suitable candidates for subsequent bioremediation and degradation assays. What's more interesting, we were able to isolate a novel fungus species belonging to the genera Fusicola, and this genera is closely related to Fusariums, and this Fusicola species is currently under description. Next, we wanted to compare the community structure in the 2 different soil profiles. So when we take a look at these Venn diagrams, we can see that the community was quite dissimilar when we compared the two profiles, as we were only able to obtain 7 common genera between the 2 and 9 of the 77 species only. So when we dug a bit deeper, we saw that at the genus level, when we went from non-polluted to polluted soil, we obtained a drastic decrease in the number of penicilliums, which was surprising to us. On the contrary, we were able to only isolate 3 genera that were only present in pH polluted soil profiles, but not from non-polluted soil profiles, which are Curvularia, Didymella, and Cladosporium. Next, we wanted to evaluate the contribution of each of the 5 culture media that were used in this approach. As you can see, Fast Fung, which is an in-house culture media, was the most performing and allowed at the same time the isolation of the novel species. Nonetheless, when we look at this Venn diagram, we can easily see that each of these 5 culture media was able to uniquely support the growth and allow the isolation of some species whose isolation was not possible using the other 4, which lead us to the conclusion that combining all 5 is crucial to allow the characterization of the soil microbiome. Next, as I've said initially, we complemented the Culturomics approach with the current standard, which is the ELIAS metabarcoding or targeted metagenomics. And using this approach, we obtained at the higher taxon levels, at the phylum level, an abundance in Ascomycota, which dominated the analyzed soils, followed to a lesser degree by Basidiomycota, which is a common finding and aligns perfectly with the other findings of other studies that studies the soil microbiome. So I'll go to something a little bit more interesting. When we ran some alpha diversity metrics, especially the Shannon one, we saw that non-polluted soils were much more diverse than pH polluted soils. Well, this isn't like something we didn't think about, but it was nice to see it. So the pH pollution impacts heavily the fungal community structure. Then at the family level, we also, when put aside the unidentified families which were abundant, we can see that Nectariaceae followed by Aspergillaceae were the most dominant families, which aligns with the findings of the Culturomics approach. At the genus level, Fusariums, Alternaria, Penicillium, Tetracladium, Darksidea, and Aspergillus were the most dominant ones, which is also similar to what we've obtained using the Culturomics approach. But here we need to clarify one thing, is that we can't directly compare the profiles obtained with metagenomics with the ones obtained with Culturomics. Since metagenomics here relies on the number of reads, which means their relative abundance, on the other hand, the Culturomics relies on the observed species richness, which means the number of species per taxon or genera. So for example, for the Nectariaceae, which came first here, and then Aspergillaceae, inversely to what you observed here, it's linked to the fact that the number of species belonging to Aspergillaceae were more abundant in Culturomics, but using metagenomics, for example, you can have like one species belonging to the Nectariaceae which dominated the soils and the number of reads, results in these different profiles. So to compare them, we need to compare a top 10 or top 20 profile like we did, and we need to see if they were present. Next, we constructed a phylogenetic tree with the top 100 ODUs, and we've highlighted the top 10, which contributed to most of the reads. In fact, we can see that some of them were differentially abundant in one of the 2 soil profiles, which led us to dig a bit deeper, and we ran some LDA analysis, and we saw, in fact, interestingly, that also Penicillium using this approach was associated more likely to a non-pH polluted soil profile. So of course, an additional number of samples will be required to confirm the speculation, but it gives us like a good indicator. So alongside Penicillium, Apiotrichum, and Coniosporium were also more likely associated to a non-pH polluted soil profiles, whereas on the other hand, Darksidea, which is a novel fungal genera, alongside Sclerosagonospora, were more likely associated to a pH polluted soil profile. So before we conclude, we wanted also to compare the contributions of the 2 approaches used in this study, which is kind of the case for all studies that combine both metagenomics and Culturomics, of course for clinical settings mainly, and not environmental. So as you can see, when we go to the species level, 41 species were common between the 2. Nonetheless, 36 were not common. So we wanted to know why. In fact, 41 is really good enough and really satisfying when compared to the literature, but we can't ignore the 36 that weren't present in the comment section. And when we took a look at these 36, we saw that 64% of them belonged to the 4 genera Aspergillus, Penicillium, Tellaromyces, and Fusarium. So for any mycologist, when we see these 4 genera, it leaves us with the same conclusion that the fact that we didn't obtain much of them in the comment section is directly linked to the fact that identification using Culturomics, which is coupled to the Sanger sequencing, allow a very accurate identification at the species level. So, which is linked to the fact that we use the beta tubulin, which is crucial for the identification of the first 3, and the TEF for the 4th one. So in other terms, for the same species that are present in the soil, we're obtaining 2 different ones. One that is very accurately identified using Culturomics, and the other one less accurately identified using metagenomics, which leaves us with the fact that for the species that should be present in the comment section, we're obtaining 2 different ones. This problem resolves when we go one taxon higher at the genera level, as only 6 genera weren't present in the common section, which highlights here the need to complement the metagenomics with Culturomics approach. So to conclude, in order to unveil and unravel the soil microbiome, yeah, it's the same one. So we can use either of these approaches separately, which will leave us with a certain characterization of the soil microbiome. But the best approach is combining them both as a complementary approach, since metabarcoding alone will allow a wider and give us a broader look at the fungal communities in soil. Culturomics will allow to validate their presence, since in the case of metagenomics, they could be dead DNA fragments or non-dynamically active fungi. But in the case of Culturomics, what we obtain will validate their presence from the metagenomic approach, will allow us to characterize them very accurately because we're able to identify them at the species level with high accuracy. More importantly, it will allow us to harness and exploit them from a biotechnological point of view, since we're able to culture them. And finally, will allow us to unravel novel fungal species that we know they exist in soil ecosystems. Nonetheless, using metagenomics alone isn't enough, since it's a non-culture dependent method. So thank you for your attention.